So I will tell you a little bit on Oedema disease and the impact on the animal. You know, Oedema disease is one of these infections caused by E. coli in the post-weaning period. And in that period, several E. coli can infect the piglets. You have the enterotoxigenic E. coli and the Shiga toxin producing E. coli, which are also called virotoxigenic E. coli. So both names are used in practice and are equally well used or equally allowed. The enterotoxigenic E. coli, they produce enterotoxins, which you probably know. The LT is the thermolabile enterotoxin, and two thermostable enterotoxins, STA and STB. Whereas the Shiga toxin is produced by the Shiga toxin producing E. coli, which are only the F18 Fimbria positive E. coli. So you have two different Fimbrias that can be present in these E. coli strains at weaning. Fimbria are colonization factors. You have F4 Fimbria and F18 Fimbria. F4 Fimbria are only enterotoxin producing E. coli strains, and F18 Fimbria are present on enterotoxigenic E. coli producing strains or Shiga toxin producing strains. So the Shiga toxin producing strains can cause the disease that you know as Oedema disease. So when you isolate E. coli in the post-weaning period, then a number of virulence factors can be identified on these strains. And this slide gives you an idea of what you can find, but it can be even more complex. So you see in this study, which was occurred in piglets with post-weaning diarrhea, F4, LT, and STB is most prevalent. But that's of course normal because you're looking for strains that produce diarrhea. But some of these isolates produced F18 and Shiga toxin, of F18 and enterotoxin and Shiga toxin, or didn't have Fimbria and produced Shiga toxin and thermostable enterotoxin. So it can be quite complex, and this can be complex on farm level. So you can have many different virotypes on a farm, but even in piglets, when a colibacillary problem is there, you can even isolate 2 or 3 different virulence strains from one piglet. So this makes it extremely complicated. This is not always the case. Sometimes only 1 or 2 of these virotypes are present on farms, but on other farms it's a complex mixture of virotypes. So let's look at these F18 E. coli, which are responsible for Oedema disease. These F18 Fimbria are the adhesion factor, the colonization factor, and this colonization factor allows the bacteria to adhere to the villus mucosa. What you see here is a villi that is scraped from the small intestine of a piglet and is incubated with bacteria. And you see all these small dots are coli which adhere to the mucosa. And that is a testing, an in vitro testing we use to see if piglets have the phenotype, have receptors present on their villi to allow adhesion of the bacteria. So this is a quite old slide, 1993. And in that slide, we compared different Fimbria, K88, that was the old name, which is now F4, and then P987, which is F6, K99, which is F5, then the F41, and the F18, which was only discovered later in piglets. And you see that the ones that are pathogenic in weaned piglets, F4 and F18, show adhesion of at least 50 to 60 bacteria per 250 micrometer villus length. So there is a sufficient adhesion needed, sufficient colonization needed of bacteria to the mucosa in order to be virulent. And the ones that show weak adhesion, the P987 or F41 or K99, are not pathogenic in weaned piglets. So F18 and F4 are the ones that are pathogenic in piglets. And if we look in more detail to F18 fimbria, then we know, you see here the fimbria on the bacteria, we know that they're buildup of proteins that form a sort of chain. And at the end of the chain, thus at the tip of the villi, you have an adenosine, a protein that can bind to the mucosa that is responsible for the adhesion, for the colonization. And this protein for F18 is called FATF. And FATF is a protein that binds to sugars on the mucosa. So you have different serotypes of F18, F18AB and AC, but that has nothing to do with the recognition of the receptor. It has to do with the major subunit. And all these fimbria, F18AB and AC, bind to the same receptor on the mucosa. So we looked in our lab in detail to the receptors that are recognized, and the receptor for the FATF fimbria, the adenosine of the E. coli, is a sugar that is located on the cell membrane. And the sugar is a blood group sugar, but it's not one blood group sugar. Different blood group sugars can interact with FATF. FATF has a groove. It has in that groove this blood group sugar fits, and this is sufficient to allow the bacteria to bind to the mucosa. What you see on this cartoon is that the fimbria come very closely to the cell membrane, and that is important to remember. So in order to colonize, F18 positive E. coli have to come very close to the surface membrane. And it's not only the sugar that interacts with the mucosa, but the fimbria also has a loop, and this loop is charged, and the charge of this loop interacts with the charge on the cell membrane, and it strengthens, increases the affinity, strengthens the interaction. So, but it indicates that there is an intimate adhesion needed. And we will come later back on that because it's important for the colonization of F18 in the post-weaning period. Furthermore, what is important is that the receptors, the sugars, they are not present at birth. This is a study we did in the lab. We euthanized piglets at different ages, from birth to 12 weeks of age to 23 weeks of age, and looked at the interaction of the fimbria, the number of bacteria that could adhere to the villi of these piglets using this in vitro villus adhesion test that I showed you before. And this is the number of bacteria that are adhering. And so you see that even in 3 to 4 week old piglets, this number is still low. And it's only at 5 or 6 weeks of age that the receptor is fully expressed. That explains partly why you detect F18 E. coli after weaning, and not immediately after weaning, but mostly a few weeks after weaning. So Oedema disease is a disease that mostly occurs 4, 5, 6 weeks after weaning. And it has to do with the expression of the receptor, but also with the intimate interaction of F18 with the mucosa. So both play a role in the fact that these bacteria colonize a little bit later after weaning. So disease is occurring when the receptor is fully expressed. That indicates that an intense colonization is needed of the bacteria. So it also explains why, if you compare colonization of infection of F4 enterotoxigenic E. coli and F18 Shiga toxin producing E. coli with each other in an experimental infection model, that F4, which is binding to a glycoprotein, which is extruding in the lumen, can much easier colonize the mucosa. So you see here very rapidly after infection, very huge numbers of bacteria in the animals, and they excrete it in very high amounts, and then this colonization rapidly drops. And after 6 to 7 days, normally, if there are no complicating factors, after 6 to 7 days, the infection disappears. And for F18 E. coli, that is not the case. And if you go on a farm and you have a problem with Oedema disease, then it's no problem to isolate the bacteria 6, 7, sometimes 8, 9 weeks after a birth. So 3 to 4, 5 weeks after weaning. It has to do with the fact that this bacteria colonize much slower. So the interaction is more difficult, and it has to do with the intimate interaction. They need really to attach to the mucosa, and then in the defense, in the mucus layer on the villi, some holes should appear, some mucus should disappear, and the bacteria clusters on the villi and spreads over the villi, and that goes slow. And so if you do an experimental infection, it's not a problem to isolate this bacteria from the infected piglets 11, 12, 13 days after the experimental infection. And the disease occurs the second week after experimental infection. So the disease is induced by the Shiga toxins. And the Shiga toxins, you see here the different toxins that are produced by E. coli. So the enterotoxins, you have the LT, the STA, and the STB enterotoxin. The LT enterotoxin is a very large toxin, complex toxin, composed out of 2 parts, an A part, the active part, and a B part, the binding part. And that this makes it a very huge molecule, whereas both STA and STB enterotoxins are very small toxins, as you can see here on the molecular weight. And this also makes that these small enterotoxins are weak immunogens, and animals do not really develop immunity against these thermostable enterotoxins. This is not the case for LT. LT is a very strong toxin, but also a strong immunogen. Now, if you look at the Shiga toxin, it resembles a little bit the LT enterotoxin. It has a similar weight, molecular weight, and it's also composed out of an A subunit and a B subunit. And in fact, the B subunit consists of 5 binding sites. That is also the case for the LTB enterotoxin. So it binds to glycolipids on the cell membrane, and this shows you different animal species and humans in which Shiga toxin can have an effect on the mucosa. Just to show you the difference between pigs and ruminants and humans. So the Shiga toxin of the Oedema disease strains is a toxin that binds to clobotetraosylceramide. It's a glycan consisting out of 4 sugar molecules that are linked to the lipids of the cell membrane. And that is different from the Shiga toxins that are produced by strains in sheep, cattle, and humans. And that's also the reason why pig strains are not pathogenic for humans and for ruminants. Whereas, in fact, the opposite can be the case because pigs, they have both receptors, have the GB3 and the GB4, and the GB3 is the one that binds Shiga toxin of the other strains. So GB4 is the receptor, and the receptor is present in the small intestine. So if the strains colonize the small intestine and they produce Shiga toxin can bind to the epithelial cells and can be transported in the animal and cause systemic effects. So the toxin is present in the small intestine. There is some discussion if it's present in the colon. There is a study that says that there is no presence of the receptor in the large intestine, but it surely is present on the vascular endothelium in different tissues. So like you can see here, and it's also present on the kidneys, the tubules in the kidneys. So this is one of these studies that is already a very old study, 1993, in which they looked at binding of the Shiga toxin of pigs to different tissues. And I made the upper part of the slide very small. That's because the concentration of binding in the brain tissue, which is the brain tissue, is very low. It increases in the gastrointestinal tract, and it's largest in the other tissues. But I enlarge it a little bit, so you see here the two toxins. The VT2E is the Shiga toxin 2E, that is the toxin of the piglet strains. And the VT1 is the Shiga toxin 1, which is a toxin produced by ruminant strains. And you see that in the brain of the piglets, the receptors for VT2E are relatively low. It increases in tissues of the intestine, the stomach, the colon, the small intestinal tissues, and it is highest in spleen, kidney, blood cells, and also the islet. And that's one of the reasons why you often see oedema occurring in the islet of the animals. So this is mainly the presence of the receptor in the tissues, so most likely on the vascular endothelium of the tissues. There is another study that looks at binding of the toxin on the mucosa, and this is jejunum and ileum, and then you see that in weaned piglets there is binding of the Shiga toxin to the intestinal epithelial cells. But in that study, 3 piglets were compared, and one of the piglets almost showed no adhesion. And this is an interesting observation because it could indicate that Shiga toxin receptors are not equally expressed in all piglets, so that some piglets are more susceptible to the toxic effect of Shiga toxin than other piglets. So what is this toxin doing? It binds to its receptor, the GB4 receptor, and is then taken up by the cells. And in the cells, the Shiga toxin moves via endosomes to the Golgi apparatus and then to the endoplasmatic reticulum, where protein synthesis is occurring. There, the A and B subunit split, and the A subunit is the toxin, the really toxic part, that blocks protein production in the cells. So in fact, the toxin enters the cells, goes to the protein apparatus, blocks protein synthesis, and the cell dies. So goes in programmed cell death or apoptosis. So that's the reason why if it binds to endothelial cells, you see the endothelial cells, you see that the endothelial cells die and you get oedema everywhere. So then we try to understand in our lab why this F18 positive enterotoxigenic E. coli or Shiga toxigenic E. coli always occur the second week, the third week after weaning, and not the first week after weaning, like F4 ETEC. And I already explained that one of the reasons is the presence of the receptor, which is not there at birth, which is appearing at weaning, but not optimally expressed, and then starts to become optimally expressed 4 or 5 weeks after birth. The second reason is the intimate adhesion of the fimbria with the mucosa. F18 fimbria need to interact really with the cells of the mucosa, and normally the cells are not free. The cells are covered by a mucus layer. So one of the ideas, one of the hypotheses we tested in our lab was to see if animals are first infected with an enterotoxigenic E. coli strain, if that could make them more susceptible to infection with Shiga toxin-genic E. coli strain. And so we did some experiments, but came then to the setup that we injected the piglets in the intestine with the LT enterotoxin that is causing secretion. And we tested different dosages and chose a dose that just dilutes the mucus layer. So not really clears the mucus layer, but dilutes the mucus layer. And if we infect them 6 hours later with F18 Shiga toxigenic E. coli, then you see that the animals that first receive LT enterotoxin are excreting the Shiga toxigenic E. coli strain for a longer period than when they don't receive a toxin. So also, if you look at colonization in the intestine, the gray bars present the animals that have been first treated with the LT enterotoxin and then infected. And you can see that the ones that have been pre-treated with the LT enterotoxin show a much stronger colonization than when there is no LT treatment. And this is also expressed in the duration of excretion. So from 5 days after infection, the ones that have been treated with LT excrete significantly higher, so significantly higher quantities of the bacteria. So removing or diluting the mucus layer makes the piglets more susceptible to an F18 infection, and that explains why this intimate adhesion is playing an important role in colonization. You see here on this scanning electron micrograph the villi, and on the villi you see these clusters of bacteria. So we think that after weaning on some of these villi, mucus is removed or diluted, and then the F18 bacteria find a niche to come close to the cell membrane and start to replicate. And then the colonization spreads over the mucosa. It's not a colonization that appears everywhere, but it spreads in clusters like a bacteria that grow on a plate. So this close interaction is needed, and normally the mucus layer protects the mucosa from becoming infected with Shiga toxigenic producing E. coli. So this is the cartoon we made of that. So first F4 E. coli comes immediately after weaning on a farm, and then this produces toxins which dilute the mucus. The receptor for the F18 bacteria is available. The bacteria start to colonize, and you see these clusters occurring on the mucosa, and disease is appearing. So that's as far as we understand now the pathogenesis. There are still numerous questions about the mode of action of the toxin. Why don't you see Oedema disease occurring in all piglets? It surely has to do with the receptor expression, but also we think that the toxin and the susceptibility for the toxin is perhaps not the same in all the animals. So a second thing we are studying in our research group is intervention strategies. And one of these intervention strategies is the use of receptor analogues. So I showed you that we demonstrated that blood group sugars are capable that the bacteria binds with its fimbria to blood group sugars. So we tested in our lab when we use blood group sugars and incubate the bacteria with these blood group sugars, and then bring this to the villus mucosa, if this prevents the bacteria from adhering or colonizing to the mucosa. And you see that in this graph. You see here different blood group sugars. The A61 is the one that we showed in the cartoon, but you have other blood group sugars. And if you use them in a concentration of 1 milligram per milliliter, they can prevent the bacteria they can decrease colonization of the mucosa by the bacteria with 60 to 80%. This is an irrelevant sugar, and it has no effect. But 1 milligram per milliliter is a huge amount of sugar you need to use. So in vitro, that works perfectly, but in vivo, it's much more difficult when you have to use this high quantity of sugars. So we went one step further and conjugated this sugar to a carrier. And by conjugating it to a carrier, you can make multimers. So a carrier which is carrying different sugars on its surface. And if you use this to block the binding of the bacteria to the villi, then you see that we go from 1 milligram per milliliter to 10 microgram per milliliter in order to block adhesion. So by making multimers, the capacity to block binding to the mucosa is considerably increased. So that's a little bit the way it worked. So we went to different experiments. In the first experiment, we tested this in a diarrhea experiment. This is an in situ experiment in which different segments of the intestinal tract of piglets are perfused with bacteria. Here you see a toxin negative F4 strain, and here you see that's a toxin negative. So if you bring in fluid in the segment, then fluid is absorbed. If you bring in an F18 AC strain that produces enterotoxins, then the fluid is secreted. So these are the two controls. So normal situation, diarrhea. So if you add now the sugar, then you can increase the concentration, and the more you increase the concentration, the lower the colonization is, and the lower the diarrhea is. And the optimal concentration in this experiment is a concentration of 8 microgram per milliliter of the blood group sugar. So we thought that with this concentration, we can go to experimental studies. So we tried that in experimental infection studies, first in our lab, and we added the multimeric sugar in fluid or in the feed in the concentration I just mentioned. And if you do that, you see that there is a decrease in colonization with approximately 2 log. And also the duration of excretion is decreasing. So it doesn't prevent really the colonization, it diminished the colonization. And in this experiment, there was disease, Oedema disease occurring in the control group, but not in the treated group. So we went a step further. We went to farm level, and on a farm level, we did experiments adding this multimer in drinking water or in the feed. And if you do that, you see that you can decrease the shedding of the F18 E. coli, and that you can also decrease the shedding. So in these experiments, we also showed that the growth performance of these animals is improved when you treat them with the sugar in the feed. I don't have that in the slide. So the second part of the strategy is vaccination. Vaccination against with an detoxified toxin. My colleague Bruno Goders did such experiments already in 2012. So if you vaccinate piglets with this toxoid, to which an adjuvant is added, then the animals develop a nice immune response against the toxin, which you see here. That is the blue curve. This is the control animals. And if you then treat the animals with purified toxin, you challenge them with purified toxin around 30 days after the first immunization, then you see that immunized animals remain completely clear of symptoms, whereas challenged animals develop symptoms of oedema disease, oedema in the eyelids, neurological signs, also in the stomach, lesions could be observed. So this showed that this toxoid vaccination was very efficient in preventing infection. So the ST toxoid immunogen is safe. It induces a good antibody response after immunization, and immunization with the toxoid protects piglets from development of neurological signs of oedema disease.